So thank you for inviting me. I'm going to talk about the seagrasses trapping plastic, surprising ecosystem services. So Posidonia oceanica is a seagrass species that's endemic to the Mediterranean Sea. It forms extensive meadows from half a meter to 40 meters of water depth and are an important part of the coastal ecosystem. This map shows the distribution of the meadows in the Mediterranean. The total area covered by Posidonia oceanica is 1.2 million hectares. And the estimated regression of the meadows amounted 34% in the last 50 years due to multiple stressors such as climate change, anchoring, or pollution. So a quarter of the goods and services have been lost. So what are these ecosystem services and benefits of seagrass meadows? So seagrasses have long leaves, so they decrease the water flow and trap particles and improve water quality. So this decreases turbidity, for example. The meadows also produce oxygen and absorb CO2, thus mitigate climate change and increase CO2 concentrations. The meadows also provide nursery and refuge for many species, so increase biodiversity and support fisheries production. Finally, seagrass meadows provide mechanisms also for coastal protection. And how this happens? So, as a temperate species, Posidonia loses the leaves in autumn. So these leaves are washed by currents and by waves and accumulate on adjacent beaches and rock beds. These leaves on beach floors attenuate wave energy, protect the shoreline from the impact of waves, so thus they prevent coastal erosion, and also they provide dune vegetation with nutrients. This is an example and a sketch of the Posidonia oceanica. So Posidonia has these long photosynthetic leaves and a leaf base called shed that is fibrous and that attaches the leaves to the rhizome. As I said, the Posidonia loses the leaves in autumn that are washed by waves and current. And when this happens, these sheets that are very rich in lignocellulose undergo mechanical erosion, and those fibers are the lazier. Those fibers intertwine to form ball-shaped agglomerates, which are called Neptune balls, sea balls, or agagropila. That comes from the Greek, which is agagros, that means wild goat, and pila, which means ball or fur, that refers to the resemblance with goat regurgitation. And these balls are washed ashore. So we wanted to know what's the role played by these piles of vegetal remains, both the balls and beach rocks, in trapping and extracting plastic debris from the sea. So our work is based on the apparent mismatch between plastic debris accumulation in the sea surface and the annual emissions of plastics in the oceans, as suggested many years ago by Jenna Jambek. The accepted or the widely accepted explanation of this mismatch between what's entering in the ocean and what we find floating in the ocean is that the explanation is that most of the plastic has ended up in the deep sea. The deep sea is the sea. However, debris circulation dynamics in coastal environments might provide a better explanation for that. And in fact, Lauren Leverton in 2019 suggested that a large part of the plastics that have been released into the marine environment are still in the shoreline. And he suggested that because he saw that the plastics that are floating offshore are quite old. So this might mean that the plastics are stranded, are buried, and undergo episodes of capturing and resurfacing in the shoreline and in the coastal environment. So the Mallorca Island in the Mediterranean Sea is an optimal site to address these issues because both of the extensive meadows of Posidonia and also because it's a place where high accumulation of plastic occurs. So we collected both balls, Neptune balls and Agricropilas from the beaches, and also leaves of Posidonia. And then we measured, we sieved, and we inspected all the plastics we found in these two environmental matrices. We also analyzed chemically the plastics with the FTIR to know the polymer type. And these are the results that we found. You can see in these pie charts the size of the plastic we found: microplastics, mesoplastics, and macroplastics in both balls and leaves, and the type of plastic that we found. And as you can see, for example, in the left, with the balls, you can see from the pie chart that most of the plastics in the balls were mesoplastics and macroplastics, with a large contribution of filaments and fibers, and then fragments. In contrast, loose leaf were characterized by a major abundance of microplastics and mesoplastics, and fragments and pellets. In this table, you can see the polymers identified by FTIR together with the density of each polymer in relation to seawater density, and their abundance in the balls and in the leaf. So if you look, you can see that polymers in leaves are principally polypropylene and polyethylene, which are polymers that are less dense than seawater. What means that polymer less dense than seawater is that a polymer that floats. In contrast, polymers more abundant in balls were polyvinyl chlorides, such as PVC or PET, and also polyamide, which are polymers denser than seawater. What does this mean? That are polymers that sink, have less buoyancy. So this means that we find some kind of sorting of plastics by density. Those plastics denser than seawater got trapped in the balls, while those plastics less denser than seawater were those caught by leaves. So here in the right, you can see different graphs. In the upper one, we show the relative abundance of plastic polymer in total plastic waste generated. This data is from Roland Geyer. Below you can see floating plastic debris in the Mediterranean. Data is from Giuseppe Suaria. And then we have our data, which is plastic in loose leaves and plastic in Agricropilas. And the red line marks the plastic, the seawater density. And again, you can note the higher abundance of high dense polymer in the balls, such as PET, such as polyamide, and such as PVC. So their negative buoyancy may have favored their accumulation and aggregation with the natural fibers in the middle. Polyethylene is still a significant proportion of the plastic trapped in the balls. Polyethylene debris might have reached the seabed due to collision with the seagrass leaves, or also because of biofouling. Indeed, seagrass beds are known to promote the deposition of sedimentary particles and to promote the resuspension of particles because of the canopy. So, in contrast, plastic debris found in loose leaves are those traditionally found floating in the open sea that were probably transported by surfer currents and wind waves and were washed ashore at the same time as leaf litter, as leaves. So this is a representation of the processes that remove plastic from the marine system in seagrass meadows. The trapping effect of canopies might create soft bottom accumulation of plastics. Then agitation by sea motion aggregates plastics and these lignocellulosic fibers intertwines and holds together fibers and plastic debris, forming these sea balls. Then sudden change in sea motion, for example, because of storms, might cause repetition collisions of these balls with the sea floor, and this induces the formation of an outward bend shell that's called the hardening of the Agricropila. This in turn might inhibit the addition of more plastics in the sea balls. And then when this storm creates high waves, these balls are released and washed ashore by sea waves during stormy conditions. What's also interesting is that these plastics that are in wracks, in loose leaves, might undergo photooxidative degradation and fragmentation because of ultraviolet light. And these fragments might be eventually washed to the ocean again. However, enclosure of plastics within sea balls protects them from exposure to the ultraviolet radiation and thus from breakdown into smaller sized pieces. So, in result, our results show that the seagrass meadows, besides all the known goods and services they provide, they are also able to counteract marine plastic pollution. And thank you very much.
It's a real pleasure to be here today and talk about some of our research to understand more about the effects of climate change and beach plastics for sea turtles. And so what I'll do is first spend some time talking about accumulation of plastics in sea turtle environments, and then how these plastics affect turtles across life stages before focusing on hatchlings and how they're affected by temperature, which is critical for their sex determination. And finally, talking about some of the actions that we might be able to take at a local scale to address these effects. So firstly, if I can move my slide. So firstly, the effects of plastics are visible across the world and even on the most remote of uninhabited islands, such as these in the middle of the Indian Ocean. And our studies have shown that the debris which is accumulating on these coastlines, which include a lot of sea turtle nesting environments, is dominated by plastic. In particular, plastic bottles, fragments, polystyrene flip-flops and bottle caps. And the types of plastics vary slightly between the open beach strand line, such as the photo on the left, dominated by plastics, bottles and fragments, and then the critical upper shoreline where sea turtles tend to nest in a lot of beaches under vegetation or close to vegetation. So our work has shown that plastics are affecting turtles across their life stages, but is most critical for onshore, where we see the debris accumulating for adults and hatchlings. And these are the adult females as they emerge from the sea to nest on beaches, and then hatchlings during their incubation period of about 60 days. And then when they emerge from their nests, which are up to 60 to 70 centimetres deep in sand, to cross the beach and onto the sea. And we've shown that plastics are affecting turtles across the world at these beaches. Not only is plastic ingested at foraging grounds, such as all of these foraging grounds shown for green turtles across the world, but green turtle diet is also related to sea temperature and so affected by climate change and warming. And plastics have been accumulating in vast quantities, such as this beach in Cyprus, indicated with the yellow circle, with over 45,000 particles per cubic metre, in the Pacific Ocean, in the Galapagos, and in the Indian Ocean. So these are just three examples of nesting rookeries where plastics are accumulating in vast quantities. And they affect turtles across life stages, even thousands of kilometres from plastic sources. So you can see hatchlings which become trapped by macroplastics on the surface, as well as by plastics in the nesting chamber itself as they're trying to emerge. They can't dig their way out because they're trapped by plastic, through to very young turtles at developmental feeding sites, such as this young hawksbill, which couldn't feed anymore as fishing line had trapped its beak close to its left front flipper, and then also causing almost kind of cauterization of limbs and amputation as fishing nets wind themselves around turtle flippers, for example. So this is a stranded green adult female that had washed up on shore dead. And there are fundamental links between climate change and marine plastic pollution, and they are synergistic stressors. So I'll just talk about climate change and warming of the sea and the sand temperatures first, and then talk about plastics. So how is climate change affecting sex of hatchlings, and what do we know about it? So temperature-dependent sex determination applies for most reptiles and also some species of fish, whereby there is no inherited sex chromosome in their DNA, but the local environment, so in this case temperature, influences the sex ratio of hatchlings. So that critical temperature phase is during the day 20 to day 40 of their 60 to 70 day incubation. And this graph shows a really healthy temperature for sea turtles in the Indian Ocean, whereby the red line at 29 degrees indicates where the hatchling sex ratio will be balanced. So 50% approximately of the clutch emerging will be females and 50% will be males. And then as the temperature increases, a greater proportion of the hatchlings emerging from a clutch of eggs will be female. And if the temperature lowers, so during winter time, there will be more males coming out of the eggs. So you can see that most of the year at this site in the Chowdhury archipelago, the temperature is low, so it's a male biased hatchling ratio. And so we monitored sand temperature at another site in the Caribbean in St. Eustatius, where a very low success rate of hatchlings was reported. So perhaps only 20 to 40% of the eggs were successfully hatching. And so we looked at the reasons for this at a beach with green turtles, hawksbills and leatherbacks nesting. And we were able to see that there was seasonality in temperature. So each species was affected by the temperature during the peak nesting period and incubation period. The incubation temperatures were extremely high, so way above that red line indicating the pivotal balanced sex ratio. And all three species of sea turtle were female biased for at least the past century. And this affected, as you can imagine, the primary sex ratio directly, so that about 50 to 75% of all of the hatchlings emerging earlier and late in the year were female, and over 90% of all of the hatchlings emerging during summer months were female. The lines at the bottom of each of the plot show the relative nesting seasons for each of the species. So the hawksbills, which is the dashed line, which are nesting in spring and autumn, had a much higher proportion of males emerging because their nests were incubating during lower periods of lower temperature as well. So what will happen in future due to climate change and also any other warming from synergistic stresses such as plastic? So at this site, it will be 100% females predicted by 2070 using ICOAD's data. And hawksbills will, by 2070, will be 100%, and greens and leatherbacks will be 100% females by 2050. So there's a dismal kind of message for the future of sea turtles at that site and many others in the world. So how is plastic affecting heat transfer? So we have ongoing field trials to investigate this question at remote beaches in the Indian Ocean, and also starting working in the Mediterranean also, where we buried plastic microbeads. So one of the common plastic polymers, polystyrene, that I showed in the earlier graphs, in surface sands. And so we have investigated the depth profile of microplastics down to 60 centimeters at these sites, And we can see that plastics are sorted throughout that sand column, but most of the microplastics and microbeads are in the top 0 to 4 centimetres. And so we applied plastic treatments using information from Emily Duncan's study in Cyprus, which has been the most polluted Mediterranean beach reported at a sea turtle nesting site, and 10 times that value as well, 685 grams per square metre. And so far, we've had inconsistent effects of microbeads on temperature. So this plot, this graph shows one of the areas where we applied different treatments, and the left two data points show you the results for controls. So 28 degrees was the median temperature, and then plastic at 1 times value and 10 times concentration on the right. You can see there's no significant effect of plastic on temperature at that site. But then at another site, you can see that the 2 plastic treatments appear to decrease the temperature for that site for turtle nesting conditions, which would suggest that plastic, in that case polystyrene, could decrease the temperature of sand and therefore perhaps balance the primary sex ratio of hatchlings emerging, but just for that polymer. So we also looked at all the other data, and it's very clear that environmental variables are greater, the variation from environmental variables are greater than the effects of plastic. So environmental variables change the temperature of the sand, such as rainfall and also wave action and high tides during spring tides. So in order to exclude the environmental variables, we then have designed some laboratory trials to study the effects of plastic in a very clearly defined temperature and relative humidity, and excluding all other environmental variables. And we selected 3 most common polymers to test. So that's PET from plastic bottles, HDPE from plastic bottle caps, and polystyrene microbeads. And we're seeing polystyrene everywhere, as you can see on the 3 lower photos. So this laboratory trial will inform beach clean strategies, and the preliminary results, which I'm not showing here because they really are preliminary, and we need to check that those results are valid. Those results are indicating that the 3 different polymers have contrasting effects on temperature. So some of the polymers increase the temperature of the sand and other polymers decrease the temperature of the sand. So if you watch this spot in about 6 months' time, we'll be able to present those results. So addressing female skew in hatchlings has already commenced by a number of different studies around the world. We've been able to demonstrate that different beaches have contrasting temperatures even within 1 kilometre of each other, with cooling effect by wind and cliff. There's consideration of natural shades such as vegetation that can adjust the temperature of the sand, and there's experimentation with low technology, low cost and available shade materials such as white sand, cotton and palm leaves to see whether you can easily adjust temperature of the sand at a local level. And it is possible to cool incubation temperatures as you might imagine. Shading can reduce temperature by just 0.6 of degree. Relocation to cooler beaches has a much larger effect. So these graphs show you that relocation from Oranje Bay Beach in the west side of St. Eustatius to Zeelandia Beach in the east side can reduce temperatures by just under 2 degrees. So that has a huge effect for sea turtle incubation and sex ratios. And watering and vegetation has also been found to be successful. But each beach is unique. So whereas we have been able to start these experiments and these conservation measures at St. Eustatius, it's important to monitor individual beach conditions before any conservation actions are planned. And so it is possible to target methods for beach management. and groups around the world have started to artificially cool nests, relocate clutches, and also do targeted beach cleans to remove plastics. And our experimental research investigating the effect of different polymers on temperature will also inform beach cleans in future to select the different plastics as priority plastics to remove from the beach. And finally, just to leave with a note that hatchlings are carried in ocean currents for years before they settle at developmental habitats, so up to 8 years or more. And threats during that time include plastics, but also climate warming, fishing, and shipping traffic. And the graphs on the left show plastic density in open ocean, and in the lower graph, global warming. And then the larger graph shows the movements of sharks and leatherback turtles and loggerhead turtles, and how they overlap over those critical hotspots of anthropogenic stresses for turtles. So there's a lot of work going on at the moment to link movements of turtles during the different life stages to threat levels. So I'll just leave with a personal note that everyone here can try, hopefully, to refill, reuse and recycle. And thank you very much to everyone for being here, joining us today, and I look forward to discussing more with you. Thanks.
Hello everyone, this is Gawsia Wahidunnessa Chowdhury from Bangladesh. I'm really honored to be a part of this ZSL Symposium. Today, on behalf of my team, I'm going to share some of the findings from the Sea to Source Ganges River expedition. Ganges, or Ganga, is one of the most iconic, transboundary, and also one of the most polluted rivers in the world. Rivers carry plastic waste from far inland and send it right out to sea where it accumulates. It is estimated that nearly 9 million tons of plastic make its way into our ocean every year. It threatens animal, ecosystems, and human health. How does this plastics and other litter get into the rivers in the first place? How it occurs? We were in a mission to find it out. Our goal was to really understand how much plastic is coming from the rivers to the ocean, to provide the first empirical baseline data on the sources, quantities, and flow of plastic along the length of the Ganges River system from the sea to source. It was National Geographic Society's largest women-led team. Members were from Bangladesh, India, Canada, UK, and USA. This interdisciplinary international team, working with social scientists, anthropologists, biologists, environmental engineers, technology experts, plastic experts, storytellers, and so on. And our team traveled from the Bay of Bengal to the Himalayas, collected data from the study sites twice in a year. In this expedition, we got a socioeconomic team, a water team, and a land-based team. We have used rapid assessment methods, circularity assessment protocol, and we applied a range of new technologies. We have interacted with the people who entirely depend on the Ganges. We tried to understand the connection between people and plastic. Following community suggestion, we developed an active learning-based environmental education program, Teachers for Planet Earth. A novel low-cost open-source tracking technology has been applied to track the movements of individual plastic litter items. A satellite bottle tag innovated for this expedition, and we tracked the movement of plastic along the Ganges River using this technology, and findings were published in the journal Science PLOS ONE. Bangladesh is one of the first countries in the world to ban plastic bags in 2002. However, plastic pollution in the diverse aquatic systems of Bangladesh is an emerging issue. We did a review study to understand the state of current knowledge and found plastic pollution studies in Bangladesh are very limited and skewed to overall snapshot. Our findings were published in the journal Science of the Total Environment. We also studied the abundance and characteristics of microplastics in the surface water in the Ganges. We identified about 140 microplastic particles with higher concentration in the pre-monsoon than the post-monsoon season. The majority of microplastic particles were fibers and the remaining were fragments. We estimated that the Ganges with combined flows of Brahmaputra and Meghna rivers could release up to 1 to 3 billion pieces of microplastics into the Bay of Bengal in a day. Findings of this study was published in the journal Environmental Pollution. Recently, our land team published their data on the rapid characterization of macroplastic input and the leakage in the Ganges River system, and the findings were published in the journal Environmental Science and Technology. We identified discarded fishing gear as a major source of plastic pollution, and findings were published in the journal Science of the Total Environment. Methods of this expedition have been published in a Sea-to-Source method toolkits to make the information open access as much as possible. And that includes the full plans and way of building your own project from any part of the world. We have identified some key solutions to tackle the mismanaged plastic pollution, and one of the solutions highlights our current project, Gender Dimension and Job Opportunities for Female Workers who have been tackling abandoned, lost, and otherwise discarded fishing nets in Bangladesh. In this project, we have been working to setting up a supply chain for waste fishing nets, which has become a holistic solution to help ensure that marginalized coastal fisheries communities can have a future ocean with more fish and less plastic built around a vibrant blue economy. Story of microplastic in the Ganges represents a global scenario. It is an emerging crisis that connected all of us. And I hope by all of us working together, we can bring a sustainable change. Because there is a solution in people. Thank you.